Next talk, Katie Hindle, Using Science Fuel to Drive a Change, a case study involving microplastic, whale sharks, and stakeholders in the Maldives.
My name is Katie Hindle, and I'm here to present microplastics, a macro disaster, a threat to the largest fish in our seas. So I was part of the team that went out to look at microplastics within whale shark feces. The two project leaders are Julia and Alina, who unfortunately can't be here, with Save Our Seas and with University of Galway and ETH Zurich. So what did we want to do? We didn't just want to use science for our project. We wanted to take science to be able to fuel change as well. So we wanted to bring our findings to the people that could make a difference on the ground, to the policyholders and the stakeholders, so people like the local communities and the stakeholders. So what are the aims of the scientific project first? So the scientific aims were to quantify and qualify microplastics extracted from whale shark feces that we'd collected in field, and also to explore routes of microplastic exposure and the links between whale shark residency patterns and microplastic ingestion. So a little bit about the background first. So a study by Tums in 2012, this graph here showed that whale sharks do two kinds of diving profiles. So they have very short bounce dives, so where they move up and down very quickly, and then they have longer dives as well, where they spend a longer period very, very deep. And they have post-dive longer surface intervals, which are thought to be for thermoregulation. So this is the longer, deeper dives and a longer surface interval. And tagged whale sharks in the Maldives have been found to do both types of dive as well, so short bounce dives and then longer dives with longer surface intervals as well. A bit of background on the area. So we collected the whale shark feces from South Island marine protected area, and a study by Copping in 2018 found that whale shark aggregations globally were found in areas that had three different bathymetry characteristics. So they were found in areas that had a shallow reef, which is was used for thermoregulation. There's a whale shark in there. Areas that were close to deeper water, and then areas that had a very steep slope as well. And this is South Island marine protected area, and it had all three characteristics. Now, Maldives Whale Shark Research Programme are an NGO that was set up in 2006 in South Ari, looking at the whale sharks in that area, and they found that the predominantly juvenile males, however, they were there all year round, which is quite significant. They weren't seasonal. So they lobbied with the government, and in 2009, this area became a marine protected area. 10 years on, there's still no policies or policing in place, so there's still anthropogenic threats to whale sharks in this area. So why conserve whale sharks beyond ecotourism? They're charismatic and they're culturally important. They've been around longer than the dinosaurs, so if we preserve them, we're preserving a piece of evolutionary history. They're very important economic-wise in the Maldives. So a study in 2013 by Kagwa found that whale sharks bring in annually about $9.4 million just to Maldives alone. They've got a wide-ranging habitat, so from coastal areas, deep oceans, pelagic. So if we manage to preserve them, then we manage to preserve many different species that live in such a wide habitat as well. And they're a great umbrella species, so they're also prone to this plastic pollution through microplastics. So they're a great species to be able to talk about, spread education awareness and stimulate action. So studies in the past, microplastic studies in the past using megafauna have been opportunistic stranding, so which means a very low sample size. So for example, this humpback whale was found with 45 microplastics in its stomach. Other studies done on tissue and muscle samples looked for plastics and plasticizers within their tissue as an indicator for microplastics. But plastics and plasticizers are also free-ranging in water, so it doesn't actually provide evidence that it's because of the ingestion of microplastics that's how it's getting into the tissue. So this is where our project hopes to provide this evidence. So Julia, who's one of our project leaders here, she identified the opportunity back in 2015 when she came out to work with Maldives Whale Shark Research Programme, and we started collecting the fecal samples. So she realized that we could actually use the fecal samples to test for microplastics. We started collecting samples in 2015. This is our Maldives Whale Shark Research team here, this is Iru and Shamil. and we continue the efforts now to collect fecal samples when we can. So to date, there's 10 samples collected. So how do we sample these processes? This is Alina, this is our other project lead. So she extracts the microplastics from the feces, and then she puts them under a Raman spectrometry and uses that to be able to identify the different types of plastics. So to date, we've analysed 5 out of the 10 samples from 5 different whale sharks. Some of these whale sharks, certainly Fernando, have been around since the start of the NGO in the Maldives in 2006, and they've all been found to have microplastics in the fecal samples. The red numbers here are the amount of microplastics per sample. And the types of plastics we were seeing, so 90% of the plastics we're finding are fibres and 10% are hard fragments. And the colour tends to be quite dark, so black and blue samples. And these are the 6 different types of plastic we've found so far within the fecal samples. So where do we think these microplastics are coming from? So the fibres we think are coming from fishing gear. There's a picture here of one of the whale sharks in the area with a fishing line free floating in front of it. And also from wastewater, so a lot of the fibres were from clothing. So if you think about when we use washing machines and we discharge that water, it goes into the ocean. The ABS plastics come from computer parts, solar panels, that kind of thing. And also PET, which is the harder plastic, so bottles, canisters. And these all are found on our beach cleanups and washed up as marine debris. So what does it mean for the whale sharks? We don't have studies specifically for whale sharks. We've not found any washed up or no samples have been done, but they are a fish, so we can use fish as a comparison. This study by Pettit in 2016 on sea bass was for microplastics and how they affect their intestinal gut. So this is the control sample with a section of healthy gut here, so no microplastics. This is untreated PVC, so non-polluted microplastics, and you can see the started to degrade a little bit. And then C, which is polluted PVC, so polluted microplastics within their diet, show severe alterations to the intestine. And this causes things like they can't digest their food properly, that causes blockages, and generally the health of the fish declines. So we think that's probably what would happen to whale sharks as well. So how does it end up in their diet? There's 3 different ways. So through plankton, when plankton ingests microplastics, these little green dots here are all microplastics and the plankton are eating them. Through direct from the water and through particulate organic matter as well. So whale sharks filter a huge amount of water. There's 3 pathways of transference to the whale sharks. So what did we do on our field trip? We had a 10-day field trip in April this year. And the objective was to find out how the microplastics got into their diet. So we picked the southern area of the Maldives and all the hotspots that we knew whale sharks were in, we picked for our samples. So we used net samples down to 200 metres to look for microplastics in plankton and seawater, and we also used a Niskin bottle to collect seawater samples as well. This was then filtered to look for particulate organic matter. We did day and night profiles, so we wanted to, we know that plankton move up and down day and night, so we wanted to do the dive profile, so we did day and night sampling. We did light trap samples, so we knew where whale sharks were feeding at night time at the surface, so we did light trap samples there, and we also did surface trawling, so in shallower areas where we knew whale sharks feed, but we couldn't drop the net down deep, we did surface trawling. So what was our analysis? So from the plankton net samples, we could look at microplastics and plankton in the water at different depths, as well as microplastics within the plankton. And then seawater particulate organic carbon samples, this we could look at microplastics in the seawater at different depths and the amount of particulate organic matter. So this can tell us if how the whale sharks are being exposed, and also if the whale sharks are following the dive profiles of the plankton. The second part of the study was to bridge the gap really between science, society and policy. So we did lots of community outreach and stakeholder engagements. These are the 2 different aims we had as a second part of the study. So the first one was to local community. So this was all about engagement. It was really interactive, fun for the community. We did lots of school talks and school chats with the local community, and we held like fun games for them to do. This is just some pictures from our community days that we did. So we explained what we were doing, why, and how they could help. The second part, we identified 3 key actors who all contribute or are affected by whale sharks and plastics in the Maldives. So scientists, tourism sector, and policymakers. And we wanted to give them a pre-workshop survey to look at their issues that they were having. And then in the workshops, we could look at problem framing and solution finding. So the scientists, tourism sector and policymakers all came up with an action plan from these workshops. We had a lot of discussions and we looked at their issues they were facing. And there was a lot of overlap actually with what they thought they could come up with an action plan. So education and awareness was the first one. So all three key actors came up with this as the first thing that we could do. The second and the third one was to monitor and fine inappropriate waste management, which is quite tricky in the Maldives because facilities and infrastructure is very hard there. So what are our next steps as a study? So obviously we need to continue to analyse the different samples, but we're also going to compile a report and a publication including the state of the issue of plastic pollution in the Maldives as we found it from our trip, what is currently being done there, and then the action plan that the key stakeholders came up with tangible goals. Thank you for listening. Thank you to everybody that was involved in our project, the team in Maldives, Save Our Seas, and ETH Zurich, and Go Away at University. Thank you.
Thank you, Katie. Any questions?
You can just shout if you want.
Sorry, maybe I missed that part. How much microplastic do you think a whale shark, another whale shark, can kill?
We haven't estimated that yet, no. So the sample, we just did the project in April, and it took a couple of months to get all the samples home. So Julia and Alina are finishing their PhD this month. So as yet, we've not analysed the actual samples from the seawater, but we have started looking at the fecal samples, which we've been collecting for a few years. So as yet, we don't know. So we can look at like the amount that they're defecating out, and then we'll have a better.
You haven't done different yet?
Not yet, no, because we haven't analysed the samples from the seawater yet. Okay.
Yeah, I was wondering if you also go for or ask for help from other companies like for divers and snorkelers in the Maldives, because I guess there are a lot of them, to collect feces samples.
Oh, collect the fecal samples?
Yeah. Do this and send it to you or not?
At the moment, it's just opportunistic. When they go to the toilet and we're there behind them, we can catch it, but it's very few and far in between. We've been there since 2006 and we've got 10 samples, so it's very, very rare that they do it when you're actually behind them and you can catch it. So, Hi.
Hi, Katie. Yeah, thanks for the talk. I wondered if you had any plans to look at any of the persistent organic pollutants that are attached to some of these microplastics.
Yeah, it's one thing we have thought of as well. So we were going to look at POPs, but at the moment we're still trying to just get the analysis done. So as soon as the girls are finished with their PhD, and we can start like looking through the analysis properly. But yeah, it's something that we've thought of for future projects as well.
Awesome.
Yeah. Okay.
Good luck.
Thank you.
Thank you, Katie, for the interesting speech.
Okay. Thank you.
So next speaker is Stefano Moro, and the title is Abundance and Distribution of the White Shark in the Mediterranean Sea.
Hello. Okay. I'm Stefano Moro from University of Rome, and today I want to speak to you about Mediterranean white shark. So first of all, I want to explain why for us, me and all the people that collaborate with me on this study, it's important to speak about Mediterranean white sharks. Then I want to explain the main objectives of this study, how we collected the data, and finally how we tried and estimated the standardized abundance trends for the Mediterranean population of white sharks. So first of all, we had a lot talks about white sharks, but known from the Mediterranean Sea. So you can understand that we have a very huge ecological cap of knowledge for this species from this area. This is caused mainly for the rarity of the species in the area and the peculiarity that there are no aggregation sites already identified from the Mediterranean Sea. There is a scientific literature, but it's mainly focused on historical sightings and reported sightings. So just a few analyses try to do some quantitative analysis on this sparse data. The consequences are that we don't have enough information at the moment about this particular population, and the conservation status that now is just uplifted to critically endangered is not supported with quantitative analysis and results. So probably the conservation, the actual conservation and management measures are not so effective in this area. So our objectives were to try to reduce this gap of knowledge as much as possible, building the most comprehensive database of Mediterranean white sharks opportunistic sightings. And then carrying out demographic evolution of the population through the estimation of abundance and distribution patterns. So we collected all the already available data from the literature, integrating it with the existing databases like the MEDLAM, Mediterranean Large Small Branch Monitoring, and then added some few records, recent records coming from social media, newspapers, or websites. In this way, we collected 773 different Mediterranean white sharks opportunistic sightings. Here you can see the temporal distribution of the records. But for our analysis, we reduced the temporal range between the 8060 for our analysis of temporal trends between 8060 and 2016. So only 744 opportunistic records. I'm saying opportunistic because there are no information about sampling effort or observation effort related to this data. So if you want to try to estimate trends starting from this data, you have to create an index of the observation effort. So to do it, we divided the Mediterranean region in 2002 different coastal regions, assuming that there was a proportionality between the human beings inhabiting the area and the number of possible observers. So starting from this, we collected data from population census data and fitted the GAMS, generalized addictive models, to obtain annual values for the human population in the Mediterranean area. Those are the results. This actually is the difference between the population in 8060 and the population in 2016. You can see that the change is not with the highest increases in the western part of the Mediterranean Sea. So starting from this standardized framework, we fitted also other GAMS models to obtain the trend, aggregating both the observation effort and the sightings in spatial strata. We used two different resolutions, one with a higher resolution like the geographic subareas of the Mediterranean Sea, the GSAs, and the other one with the FAO divisions. So we use the observation effort as an offset to obtain something similar to a CPUE, so number of sharks per a fixed number of humans. But in our modeling exercise, there were like a trade-off between the spatial resolution and the temporal resolution. So using the GSA, we had been able just to estimate an overall spatial and temporal effect. That's are the results. As you can see, the overall temporal trend showed us a very steep decline started in the last 30 years, while in spatial terms, we could identify main hotspots of presence, and like the Sicilian Channel that is already known to be, I don't know, an hotspot of white sharks in the Med, and the Balearic Islands. But so reducing the spatial resolution, we had been able to obtain a singular trajectory for singular sectors. This is the result using the FAO divisions. As you can see, in most of the sectors, we detected declines in the last years, but looking more precisely, we found declines that started earlier in peripheral sectors, such as the Balearic and the Marmara Sea, and with higher magnitudes of decrease, while in central sectors, the decline started later, more recently, and were with magnitudes of lower intensity. So we hypothesized that could be happening a shrinkage of the spatial distribution in the area. So to test this hypothesis, we divided our data in two main time bins after World War II, and just tested the differences in abundance within the different FAO divisions within the two time periods. And as you can see, with our GLM, sorry, the central sector showed an increase while the peripheral sectors decline. So to conclude, we used this pattern to try to make some ecological hypotheses about which were the possible drivers of this change. And clearly, we think that is it related to the strong increase of inshore and offshore fisheries, but maybe it could be related to a predator-prey dynamics with the bluefin tuna population. In fact, if you look at these statistics, these are the stomach contents and the fishing gear interaction. More than a half of our catches data are related to tuna traps, while if you look at the stomach contents for teleost, more than a half, maybe 47%, not more than a half, tunas. But clearly, we did this very simple assumption, so we cannot exclude that there are other factors than human population that are affecting the change in the observation effort. So to conclude, with this study, we have been able to identify hotspots of presence for the white shark in the Med. Using the human population as a proxy revealed to be a very useful tool because it's single, is really is easily available and systematic. So can be used in general for any kind of opportunistic data. We detected the population decline with a parallel contraction of the spatial distribution, and in some way demonstrated that even if you have opportunistic data that I think if you ever work with that are not the best ecological data you can use it to analyze that to postulate intriguing hypotheses about the ecology of the species that you are studying. I did it with the white shark, but I don't know, everyone can do the same with other species. And actually, I would like to try to do it. So, nothing, that's all, thank you all.
Thank you very much, Stefano. Any question?
Yeah?
Thank you very much. It was a lovely. I wanted to ask you if this data comes from catches or catch and releases as well.
I have a mixture of searches for the data. I have strandings, catch and release, catch sightings. I used everything I could find. Why are you asking that? Because?
No, I just wanted to know where the information is coming from. And do you know if there is an important area between Greece and Turkey as well for the white sharks?
In the last 15 years, I know that had been caught like more than 8 different young of the years in one bay in Turkey. But I don't have enough, we don't have enough data to try to estimate patterns for different age classes or size. Maybe for size we can try, but.
You said that you use also records from social media. You mean photos, etc. Okay. In my experience, it's very difficult to distinguish between young great whites and mako sharks. So how did you fix this problem from the photos?
Actually, I used only records that were validated by experts. So I don't know, I just trusted the person that did the identification.
Okay, thanks.
A last question there.
Hi, Stefan. I was wondering if you made any searchings using Arabic.
Sorry,
Because
what?
when you're looking at the records,
Yeah?
the fact that you have almost no records in Libya and Egypt
Yeah.
are striking. So I was wondering if you are somewhere, because you're not using Arabic, I don't know, can you increase the effort in your search for those particular areas? Are
What do you
those
mean,
real absences, or is it because you're not looking?
Clearly could be a bias related to the northern part of Africa, all the southern part of the Mediterranean coast, because the countries had few political instability problems, so actually maybe report a white shark sighting is not the main problem for a person living there. But I said in the presentation that we know that there are other factors driving the observation effort. So one of the next steps that we want to try to carry on is to find always more robust index of the observation. And we started just with the population because it was the easiest thing to do. But clearly there are many factors. Also, I don't know, just if you think to the increase of the social media in the last years, that probably boosted the number of sightings you have. Is the same.
Okay, thank you very much.
Next talk, Pablo Garcia Salinas, Baselines for Butterfly Babies Towards a Mediterranean Study Model.
Okay, hi, good afternoon. It's almost there. Thank you for being here until the end. Well, what I'm going to show you are the first results of our preliminary study in the Spanish coast that Lamna Association, with the help of Servo Search Foundation, have made during the past year, looking for these animals, the spinning butterfly ray, Gymnura altabella. Gymnura altabella is a benthic batoid that we can find in the soft and muddy bottoms of the Atlantic Ocean and the Mediterranean Sea and in shallow coastal waters. Being this distribution, the problem that we have is this animal is really sensible to the human impact. In fact, at the global scale, we saw that the population is decreasing, and in Europe and the Mediterranean is not just vulnerable, it's critically endangered. Focusing on the Mediterranean, we can find this animal in the bycatch of coastal demersal fisheries, but although it's not a targeted species. In fact, in some countries, such as Spain, the animal is protected by the law, but due to its life history and distribution, the animal is also vulnerable to the coastal development and the tourism activities that can be a threat for the future of this species. So we wanted to determine the annual presence of Gymnura altabella in a high tourism area in Spain, but also identify possible congregation areas, because in the Mediterranean, Gymnura altabella is considered to have a patchy and fragmented population around all the shore. So the detection and determination of these areas can be an issue and can be a key to the protection of the animal in the future. And also, we wanted to create opportunities for new studies, and why not future plannings for the conservation of the species. We think that this species is really big, it has a quiet behavior when approached by the divers, and it lives near the beach users, so it can be a charismatic species for future conservation plans. So to do that, we start doing some surveys in different areas of the Spanish coast. Mainly we focus in two different areas. They were quite similar underwater with wide sand extensions and also a really well preserved seagrass meadow of Posidonia oceanica and Cimolocea nodosa. But one of the areas has a high tourist presence and the other one has a less tourist presence because the accessibility to the beach and also the facilities around the area. So in each location, we developed a series of monthly surveys. We spent a lot of hours into the water doing always the same pattern to try to figure out where these animals were. We start recording, I don't know if you can see the signs, the marks in the sand, but well, are there. We started recording every mark that we found. These marks, you can call it footprints, fin prints, or body prints, whatever you want, are left by the animal while there is waiting for hunting. We just focus on the clear ones, the ones that were really well defined, and not the blurry ones. We take measures of every one of the different marks, and also we take the depth in which the mark was found. I have to notice one thing that is important is with these marks, we cannot tell the number of Gymnura altabella present in the area. We can tell that the animal is there, and also we can tell the range of size, but we cannot tell the precise number of animals. With the alive animals that we found, we do the same. We measure the animals with a measure tape and with paired laser photogrammetry, and then also we took mucus samples of the animals to future genetic studies. I have my freezer filled with mucus of Gymnura altabella, so if any one of you want to do anything with genetics, it would be great. And also we took some pictures of each one of the animals. We start doing that because we are not allowed to manipulate or tag these animals, so in order to identify them, we have to use photo identification. But soon we realized that it was really hard to remove all the sand from a 1.5 meter animal, and also we were disturbing the animals. So we started doing it by just fixing in the different spots that we can find in the tail of the animals, almost like a barcode, to identify all the animals. So until now, we found 116 clear marks in both areas. We have found a higher amount of marks during the spring months and a drop in the sighting of marks during the summer months. I'm sorry, because of the letters have moved a little. We found 32 alive animals in both areas, 12 males, 20 females. Males were typically smaller than females, and we found a high presence of males during the spring months, and females were bigger than males, and we found females all year round constantly. And also one thing that is interesting for us is we have found 3 females with signs of, where is the point? No, is the center? Okay. Yeah, you can see the signs. We think that perhaps these animals were pregnant, but we cannot assure that. Regarding this distribution, we always find the animals beside the Posidonia oceanica or the Cimolocea nodosa. Just the smallest of the juveniles were found within the Cimolocea nodosa in the small patches of sand in the middle of the seagrass meadow. We think that's because 2 reasons. The first and mainly perhaps is that there's plenty of food in these well-preserved areas of Posidonia oceanica. There's a lot of fish these animals to eat, but also if you notice the long leaves of the Cimolocea and the Posidonia sweep or clean the riptide marks that we can find in the sand. So for the animals, it's easier to hide in this clear area. As I told you before, we found these drop in the sightings during the summer months. But when we analyze the data, we observe that actually the drop in the sightings was just in the area with a high tourism level. And if we do it for a long time, we spend some time underwater, you will see clearly what's the reason. There's a lot of chains, of chain marks and anchor marks of all the boats that can stay in the surface in the same moment. So clearly the animals are not there. So just to resume, we have note the annual presence of Gymnura altabella in an area. This area was, this animal was previously supposed extinct in the literature, so, and we know right now that there's a small population of at least 32 different animals, with a small seasonal variation, more animals during the spring, and a drop in the sightings at least in one park during the summer, probably due to anthropic pressure. But if we want to move from a small pilot study to a future established project, we have to explore other areas, obviously. We have to determine the habitat use of the animals that made of the space. We want to develop hormonal analysis. We were talking right now with Universidad de Barcelona, because they are really, they have a really strong group of people working with hormonal stress, stress hormones, sorry. And we want to do it during the summer months and the entire year. And also we have to start working with the DNA and all the samples that we took. Perhaps to develop some environmental DNA tools to prove the existence of more animals in the Spanish coast. But also, talking with the people is important, and right now we are setting up a meeting with the local government of the region, because we want to share our main results, but also we want to ask permission to put some information panels about the presence of these species into its areas. And also, we want to develop a manual of good practice for the sport fishers and also the divers. And just to finish, I have to say, and I really want to say thank you to Save Our Seas Foundations, because they funded us to make our little project. This is me holding my brother. This picture was taken in the same place that right now we are doing the experiment or the research, but this picture is almost 30 years ago. Back then I have no idea that I was gonna be doing this kind of research in the future in this area. And I saw the animals there. And for me, they are always been there. And they were going to be there in the future, at least in my child mind. Now I think that, well, the animal is critically in danger, but I think that they can still be there for the future. So, thank you.
Thank you for this interesting speech. Any question?
No?
I do have a curiosity. I don't know if I missed something, but can you recognize from the mark on the sand the sex, like if the male, large male, mature males with long clasper? Can you find
If
it?
the mark is really good, is really recent. The sediment is perfect, and the rate just left. Perhaps we can do it. Okay. Now we have found marks that we think that are clasper marks, but we still have to try to analyze all the images and try to do it. But yeah, perhaps we can do it. Because there are some, when the animal was breathing, they left longitude, well, like a mark in the opposite part of the body, and you can confuse it with claspers. Yeah, but yeah.
Okay. Thank you.
You're welcome.
Other questions? Thank you very much, Pablo.
Okay, thank you.
So now we have our last but not least speaker, Letizia Sion, and she's going to talk about the Lasmobranch community in the Central Mediterranean, changing diversity, abundance, and habitat influence.
Thank you. Good afternoon, everybody. Bye. So, the Mediterranean Sea is considered a biodiversity hotspot of Lasmobranch, and the data collection by UCN underlined a high percentage of threatened species out of cartilaginous. But rebuilding replaced the Lasmobranch population is considered a high priority of key conservation for the Mediterranean Sea. So we have a big gap of there aren't standardized catch data of Lasmobranch species are available. So information to reconstruction, so especially spatio-temporal trend of these sensitive species provide have baseline, especially with the current species and an abundance in an ecosystem can be measured. So, especially in our area, in the Ionian Sea, these Lasmobranch have a negligible role in fishery common, but they have ecological function important, and especially on the role in the trophic web. So we are collected the data came from national and international research project, and the study area is the geographical subarea 19, geographical subarea FAO, localized in northwestern Ionian Sea. This area between Cape Otranto in Apulian region and to Cape Passero in Sicily region. But we collected data coming from 25 experimental through survey medits project. Then we used the experimental TrollNet, and allocated 70 holes for each survey in a deep range from 10 to 800 meters using random stratified sample. So we have obtained species diversity, and we calculated also fluctuation of abundance in terms of density and biomass standardized to kilometer square. But we want also to improve the knowledge of chondrite in our area, investigated also in particularly habitat sensible area, especially in Adriatic Sea, in Bari Canyon, and in cold water in Colaria province in Santa Maria di Leuca. So we used tools with lower impact on the bottom. In fact, we used experimental long line in depth range 180, 30, and until to 712, using systematic sampling and video system by Memolander. Using systematic samples. So we obtained abundance in density on our. So regarding species diversity, we have collected 37 chondroited species divided in 70 shark, 90 batoid, and 1 chimera, species Chimera monstruosa. And these species have a wide bathymetric range, reaching also 1,500 meters, and also a wide size range. We can see that the species has Exanchus griseus range 4 meters. Then we can see that the trend of abundance in the long time 2020 survey, we can observe a significant increasing of biomass of elasmobranch in Northern Ionian Sea, and about Galeus melastones, the more abundant species. Also, the significant increasing of biomass was detected. Also, analysis elasmobranch assemblage were carried out. So we observed the presence in the Ionian Sea of 2 distinct groups of elasmobranch. One group allocated in the northern regions and north Calabria, characterized of the lower number species, and the second group in the southern Ionian Sea. And this difference is possible due of environmental factor between the South and North Ionian Sea, but also there is also the narrowed treble bottom, so the fishing activity is lower in the Southern Ionian Sea. Then in the investigation in the sensitive habitat, we find 13 species, but there is the difference between different tool, long line, and video memolander, and also Adriatic Sea and Ionian Sea. Thank you to memolander, it is a stationary tool. We are possible to collect data on Exanchus griseus, because this species has a behavior to quiet and on the bottom. Then important is also the abundance that compared the abundance between inside collar area Santa Maria di Leuca and out area on where don't find collar area. So is evident observed that the density is more abundant for these species. These species are more abundant inside area, and also the population structure for this species is we can see that the Chimera monstruosa have always individuals, juveniles, and greater individuals, while Atmopterospinas is characterized especially young individual. So we can underline that the recruitment and spawning phase of I love the cycle is there is in the coral area. Also in the Barry Canyon, about more abundant species Galeus melasponus is we can see that abundance in terms of number and biomass is more in Bari Canyon respect adjacent area. So also the population structure is characterized more individual with greater size. So in conclusion, we can see that it's important to investigate with different sampling, so high impact and lower impact. So we can collect 1317 on the Ionian Sea. Also, these species are rare, and occasionally we have collected. The most important species are more abundant species are Galeus melastomus and Etmoptospinax, and where there is high fishing pressure, but this could be due the very deep distribution. So we find that the greater number, the greater abundance and size we are recorded in sensibly habitat like Bari Canyon and Santa Maria di Leuca, where there is a low of fishing pressure. Conservation and management of these species, cartilaginous species, require a reduction of fishing pressure and fishing money, especially in essential fish habitat, where reproduction and recruitment occurs in the Bari Canyon and Santa Maria di Leuca. So it's recommended that this FRA declared the limits of commercial triangle, but it preserve this site and allow conservation and especially the management of the, in general, several demersal resource. Finally, we can underline that fishery restricted area there is in Santa Maria di Leuca Colar area, and establishment of a FRA in the Bari Canyon, and we have recently proposed, and the institution process is in process, and we hope that our study can is the support for this establishment. That's all. Thank you.
Thank you, Letizia. Any question? So, there are no other questions. I do have a small question. Did you see any difference in sex distribution between coral and non-coral area?
No, we have sex distribution, no, but we have we have 5 especially mature females and a big fraction of females and males. And this is important to in coral in this refuge area, especially in the spawning phase of this species. Thank you.
Interesting. Thank you. Any other question? Okay, thank you very much. Bye. In the end of this session with Primo, we want to thank all the speakers for their very interesting presentation about conservation and communication in different areas with different approaches on different species. So we think it has been a very interesting session, very long but very interesting. And also the speaker have been really in time, and so we really appreciate it because it was quite long. So thank you very much, everyone, also for remaining here until the end of the session. Thanks.
Well, now we are going for the conclusion of this meeting, But before I invite Claudia, Claudia is in the room, yes, she's coming. She has something to tell. Then we will proceed with a small ceremony in which we will give the prize to the children that won the drawing competition. The children will be in the room in few minutes, but after that Claudia will tell us what she needs to tell. Where is Claudia? Here, there, where you?
Thanks a lot, Emilio. This will be very quick, I promise. I've said this before, but it will be quick. So first of all, we just wanted to point out again that we managed to raise 920 euros yesterday for the student bursaries. So that was a brilliant job. Thanks everyone who contributed and who was buying one of these amazing items we had yesterday. On the topic of student bursaries, so I hope you're all familiar with the idea of our student bursaries, that we're raising money every year for the auction, as part of the auction, that will go into the student bursary funds for the next years, and that will allow students in the future to come to the conference. So we're handing out these student travel bursaries for students to present their work. I would very, very, very much appreciate if everyone who's seeking for these bursaries really, really reads the instructions for the applications, because we will publish all the guidelines on the website for people to apply. It's not rocket science, but please stick to the requirements of the bursaries. Otherwise, there will be no bursaries. That was it, I think. I guess so. And congratulations again to everyone who received a bursary this year. I think we awarded 10, Glennis is nodding. We awarded 10 bursaries this year, so congratulations to everyone who got a bursary this year. And yes, thanks for presenting.
Well, now, welcome. We have some special guests. Traduco. Benvenuti. Siete i nostri ospiti speciali. As we made a competition in effect, we want that this meeting was devoted to the communication of Erasmus Branch Science. We worked all year not just for the meeting. We worked with the schools. We worked with children. We arranged many lessons and many activities in the schools. We worked with high school during the meeting between you. We had 7 students, the best 7 students of our region. We offered them the inscription to the meeting. They were really interested in shark science, so they were between you, and they participated to all the sessions. And now we are here because we want to give to these children the prize for their drawings. So I would like that Barbara, Ollie, Sarah will come here. We will stay down so you all can see the ceremony to give the prize, and I will take the other microphone. First of all, I must thank Professoressa Matilde Lapano, which represents the director of the school. The school is l'Istituto Comprensivo Statale Rende 4 Miglia. The name of the project was Alla scoperta degli squali, Discovering Sharks. And now, I invite here Sofia Ippolito. Come here. No, glieli danno loro. Dagli Barbara. We take a picture. Then, Ollie, are you ready? Ferrise Francesca. Take a picture, fai una foto con lei. And of course, the founder of the EEA should give the prize to the first in the competition, Mario Fortino. Bravo! See, Alex, for a picture and for giving the director of the school the certification. Okay, congratulations. And thank you to Patrizia. She arranged all the activities with the school and with the children. Thank you. Now, the children can go, but you must stay here. Diana Pasmigno. Yes. Voi potete andarvi a sedere. Terry returned home. I must say thank you to these beautiful women because they made our meeting excellent. I am very grateful to them. And like for Bruno last night, I asked to a Calabrian artist to make a special shark for each of you. If you give me 1 minute, I will bring them. Maybe, I don't know if it will be too much big. I hope no.
No worries, I'm not gonna sing.
help our guests to... Is mine? No. Where is mine? There, yes. No. Is not there? Oh, well, never mind. I will keep in mind all. Well, I must thank, and I hope you will agree with me, first of all, the EEA board for let me arrange and organize this meeting. The Centro Studi Squali and the Greece for helping me during the organization. The technician of this main hall, Franco. And all my students, because they worked hard. This meeting has been possible above all thanks to all of them, and at the end of this event, I will ask them to come here, and I would like that also them can have a picture with us. Okay? Thank you.
Well, now, it's okay? Yeah. I know all of us are very tired. Just a couple of minutes, I will be very brief. Thank you, all of you, for coming and for being with your talks, doing this conference very, very interesting. I'm going to give you just a few numbers that Emilio gave me to understand the magnitude of the conference that we had here in Italy. We had 205 delegates, without counting the ones of the University of Calabria. Exactly. So counting the students, we have 229 delegates among 30 countries, which were Australia, New Zealand, Mexico, Ecuador, South Africa, Algeria, Israel, Algeria, Cabo Verde, USA, Canada, and a lot of EU countries. So thank you very much for coming and for doing this very big. 71 oral communications, 59 posters, and about 60% of delegates were students, even if they were bachelor, master, or PhDs. So it means that the health of the shark science is increasingly and growing. So very good, very happy for doing that. I also have to thank you, all the organizers. So Greece, the Centro di Studi Squali, and also the University of Calabria for hosting us. All the volunteers, all the students that have been helping with the organization, etc. And of course, a big, big, big thank you to Emilio, because no, thank you very much, really, Emilio, for your passion, for your involving, for your kindness with everything, for your patience with all of us, asking to you everything. So really, thank you very much. Then, also we want to thank all the sponsors, and especially at CISA because always is very helpful and supporting, especially with giving funds for the student bursaries, which is always very important, that which allow to the students came here. Again, thank you everybody for being here with your interesting presentations and posters and everything. And also I want to thank, on behalf of the EEA Policy Committee, your involucration, giving us over 150 signatures on the MACO letter that we sent yesterday. So really thank you, which is in just a couple of hours having this amount of signatures, it's important, so thank you very much for this. Just a personal comment from me. During these last 2 years. Of course, we have rotation turns, and now I have to welcome new president, which is Claudia Jung, which will be the new president for the next 2 years. And so best wishes for your 2 years of mandate. And that's all. All of us are very tired. So thank you, everybody.
I remember that we will need for tomorrow and for Sunday help with shuttles. Please contact me. I will be outside in the desk for the next 15 minutes and then stop. I remember, because a lot of you asked me again, at 7 tomorrow morning from the university, at 7:10 from the round road near the McDonald's in front of the Majoran Hotel. And now, in 10-15 minutes, if you go to the bus stop, you will find the bus that will bring you to the hotel like this morning. So Majoran and Daria, then Domus, then President.
Just one thing, please, all the EEA board members, stay a moment because we want to take a picture with all the students and volunteers that made possible this one, so please don't leave.
And also the speaker with our student, one picture. Ragazzi, venite a farvi una foto.